Okay, getting my laser pointer out. So, does everybody know who this is? This is Michelangelo's David. I have a friend who's an ER doc. She's really fighting through this COVID-19 thing, and I hope she stays well. But when she went to med school, her they started out her intense lectures on kidneys with this picture, and I'm going to do the same with you. So we kind of think about, well, the kidneys make urine. Um, Her professor said uh, kidneys make urine is saying that Michelangelo sculpted David here to make marble chips. What the kidneys do is maintain the blood. You can think of this statue as maintaining the blood, its exquisite structure. And then urine is the byproduct of that process. That's not the main thing it does. It maintains the blood. So we think of blood as the statue David, chips on the floor, marble chips on the floor as the urine. So what do the kidneys do? We'll begin our discussion today, and this is going to be relatively short. The kidneys can be extremely complicated. I really didn't fully understand things until the end of my grad school career, and part of that was a lot of self study, trying to sort things out, trying to figure things out. We're not going to get into that depth. What I want you to take home is that the kidneys maintain the blood. They make that statue of David. The urine is a waste of this process. So we want you to think about how this gets done, the parts involved, and what the main purposes are or the main functions of the nephron, which is the functional unit of the kidney. When we think about the kidney, we got to think about the pathways of liquids through the kidney. And we have to remember that there's one way in, two ways out. So the renal artery here, we're bringing in blood. That's our one way in. Liquids can lead either through the renal vein, so they go through, circulate through the capillary beds, multiple capillary beds, and then come back the renal vein, or they can be taken out of the blood supply and leave through the ureter. So on its way to the bladder, leave as urine. So leave as perfectly reconstituted blood, or leave all the extras, all the waste, all the things that we don't need to make that perfect blood are taken away into the bladder. We need to think about, as we look at this, the layers of the kidney. We have an outer cortex and an inner medulla. We think about those two things, two levels. They are very important in the kidney's function. The different layers have different environments that help concentrate the urine, help conserve different substrates, and we'll take a look at those as we move through this lecture. So we want you to remember you got renal artery, renal vein, the ureter, one in, two out, cortex, medulla. We'd like you to remember that this is the pelvis where all these things come in and out and then radiate out to the capillary beds. So if we compare kidneys across the species, they all perform the same function, but they have a slightly different arrangement of the structure necessary to get this done. So we're going to focus on our cow here in the middle. Got a horse over here and a sheep over here. Sheep sort of looks like what we'd expect when we think about a kidney, but when you take a kidney out of a cow, you got to have this huge grape kind of thing. These grapes are what, golf ball size, tennis ball size lobes in there. So we have a smoother finish in the human, a smoother finish in the sheep, smoother finish in the horse. But again, all three have the one in, two out arrangement. So one in, two out. So we cut away with that structure there, and we see that each of these individual lobes has a cortex around the outside and the medulla inside. We get over here with other animals, they have a more or less continuous cortex, more or less continuous medulla going through. So it's one big lobe, whereas a cow has got this structure set up over and over again. Each of these lobes is um, each has its own cortex and medulla. Slightly different in the pig, you got that smooth outside, but you have distinct sets. Again, the cortex and medulla. Cortex meaning the outside, medulla farther in. And this helps the kidney function. Different processes occur in the cortex than they do in the medulla. If we look how a cow is set up, we have our ureters coming out. Not sure which is which. You can see on your handout 7, 6, and 7. I think this might be the artery here, the white, and then the 6 might be the vein, but I could be wrong about that. I'm going based on a structure here. More robust, thicker wall here would be the artery. Thinner wall would be the vein. So if you're laying on, we're looking at a ventral view. We're looking up through the belly button. These kidneys are tucked in right up on the back. So they're on top of all the guts, up on the dorsal side of the body cavity. We have the adrenal glands sitting up on the top on each side. They have their separate uh, blood systems going through, and then we have these multi-lobed kidneys. One's a little bit more interior than the other. So I'm guessing this would be the right one's a little bit farther back. We again got our dissection here. This is focusing on our ureter going through. You've got the collection of the urine from each of the nephrons. They get in greater tubes branching together. Each lobe has got its own drainage, and it ends up in a common drainage of the ureter to the bladder. So what we really want to get down to is the nephron. Nephron is the functional unit, the unit that does all the work in the kidney. So we want you to know your nephron parts. So this is a schematic of a nephron. You've got your cortex medulla border here in all of our kidneys. We got a cortex area here, medulla area here. We had those common collecting tubes that are going to the ureter. So if we go backwards here, we have a collecting tubule. Each of these branches is a connection to another nephron. All the nephrons come together, collecting at the end. The blood comes in. It establishes a capillary bed in what's called a glomerulus, and then the blood leaves again and goes to a secondary capillary bed. What happens? We go in here, and then glomerulus is surrounded by the Bowman's capsule. This is the start of the nephron. Liquid comes from the blood over here. It's going to be put in. We're going to have the proximal convoluted tubule. That's the first one you come to in the nephron. So nephron continues on. As it crosses into the medulla, we get the loop of Henle. So Professor Henley, wherever he or she was, first to define this, we have a loop that goes down into the medulla and then back out. As we leave the loop of Henley, we've got a distal convoluted tube. So the one closest to the front end is the proximal, the other one is the distal here. All these distal convoluted tubes empty the nephrons into the collecting tubule and go down the urethra. So what happens here is the blood comes in, you have a very leaky connection between these two. All what happens here is most of the plasma leaves. All that's left that comes back out is sludge with just enough liquid for it to move. Everything else, all the plasma, is put into the Bowman's capsule. And then the job of the nephron is to pull everything back that is useful. So you have all these different parts that pull different substances back. Whatever that does not get pulled back in ends up in the ureter, ends up in the bladder, and that becomes our urine. So the idea is that the nephron pulls stuff out, pulls it out, and then we have a secondary capillary bed here that collects all that stuff and puts it back in the blood. So the nephron, if you're looking at the location in our cattle, you have the start of the nephron. We go down loop of Henle. We go back into the cortex, distal convoluted tube, and then the collecting tubule that drains into the ureter. So this is the orientation of the nephron, proximal, distal convoluted tubules in the cortex, loop of Henle, collecting tubules starting in the cortex going to the medulla. So here's another kind of probably a better, more accurate diagram. Again, everything's labeled. We've got the Bowman's capsule. We've got the proximal convoluted, so we see a lot more convoluting happening here. We cross over into the medulla. We have the descending loop of Henle, the ascending loop. We start get into the distal convoluted tubule, and then we switch over to the collecting tubules. Each of these stems here, each of these openings here is a connection to a nephron. Multiple collecting tubes, multiple nephrons serving in the same collecting tubule. So arched up here, straight going through. Each of these sections does something different. So adding a layer of complexity to this, we have our artery here working our way through. We have our first capillary bed in each Bowman's capsule. And then we have the sludge with just the cells with just enough liquid, the sludge that's going to move through a secondary capillary bed around the nephron. You can see that nephron in here. It's surrounded by that secondary capillary bed. And this is where all the things that leave the nephron that are actively removed are put back into the blood. So by the time we get to the end of the second capillary bed here, we've reconstituted the blood, we've cleaned the blood. All the things that don't belong in the blood are now working their way down the collecting tubule into the on its way to the bladder. So we take everything out at the Bowman's capsule, all the liquid, so mostly plasma, so all the electrolytes, most of the proteins, not all of them, all the hormones, all the electrolytes, all that stuff, sugars, all that stuff moves into the nephron, and then whatever the nephron wants, it removes from the nephron and puts it back into the bloodstream. So it moves from here to here, from here to here, from here to here. We have everything moving in. So those are all the solutes of the blood, the electrolytes, the sugars, some of the small proteins, all the amino acids, all the hormones are going to get taken from the nephron. The nephron going to say, "Hey, that's good stuff. I'm going to reach in to the inside, pull it out, and throw it back into the bloodstream." So you've got coming in from the heart, going back to the heart. So let's lay this out in a functional thing. What I want to do here is stretch out the nephron. I'm going to take the Bowman's capsule and the glomerulus, and then instead of going through, we're going to lay it out and stretch it out into a straight line. So proximal convoluted tube, loop Venley, distal collecting tubule. So all the liquid comes out here, and we have effectively plasma, everything but the red blood cells and the white blood cells. All the cells and a bit of water, the sludge starts to go out, and you're going into the secondary capillary bed, which is here. By the end of it, we're going to have reconstituted blood and our urine. So the idea as we move through is that the nephron pulls out all it wants and then puts it back onto the blood side. Whatever it doesn't pull out of the nephron is going to go to the bladder. Whatever gets pulled out and moved over ends up in the heart, back in the circulatory system. So by losing the things that are not needed, not wanted, we remove the waste products, we create the marble chips, and then we create our statue of David down here, our reconstituted, perfectly cleaned blood. So what happens in this process? We've got a net movement from our nephron over to our blood. What starts this process is the needed solutes, the electrolytes, the sugar, the amino acids, the small proteins, anything that the body wants, the nephron will remove and put it back into the blood. So these are the solutes generally are moved first. Then as we get over to the collecting tubule, water moves passively over to the blood capillaries. So the nephron actively moves the solutes, say, hey, I want that. I grab it and I throw it back into the capillary. And water goes, hey, wait for me. I got to go after. So water follows passively after the fact. So again, you've got everything back in its proper orientation. Liquid moves from the nephron to the capillary. Solutes move from the nephron to the capillaries. And we got reconstituted blood here. And then we've got our urine, whatever that may be. And that can vary immensely from day to day, hour to hour, what that is, depending on our hydration state, depending on our electrolyte state as we move through. So, to illustrate this point, I want to talk about messing with your friends. A group of guys I went to high school with, we get together on a regular basis. We mess with each other because that's what friends do. And one of our friends has a friend who's a pharmacist, and I don't know how he got these, but he said, hey, I want to feed this to Bill. And he goes, well, I'm like, why? Paul is the procurer of this. I said, Paul, why do you want to do that? And he goes, I just want to mess with him. He says it's going to turn his urine bright red. So me and Paul are standing there in the kitchen, and we go, how to get Bill to consume this pill without noticing what's happening. So I said, well, let's do it this way. I took a chocolate-coated donut. I pushed the pills into the donut, and I walked out into the living room, and I said, Bill, I bet you can't fit this whole donut in your mouth. Now, Bill is a member in our group that can't turn away from any sort of gastronomic challenge. He'll eat anything. He'll try anything. He will try to do things like that. So we got him to fit the whole donut in his mouth, and he said it was after the fact. He said it's a little crunchy. Tastes a little funny, but I assume that was just because it was the chocolate coating on the outside. So we got him to consume these two um, antibiotics, and the side effects of this antibiotic as it's cleared from your body is it turns all your bodily fluids orange red in color. It's a benign side effect, but can be frightening if it's not expected or you're not prepared for it. Especially if you're on the toilet and everything gets bright red in some way, shape, or form. So we went hiking later that day. It was a camping trip in the winter. We went, and along the way, for whatever reason, Bill fell. I forget exactly why or how or whatever, but we were standing. He's like, I'm going to go relieve myself. So he walked behind the lean-to relieve himself. And he got in the back, and me and Paul are trying to hide our laughter. And Bill starts screaming, "Hey, hey, hey!" I'm bleeding, bleeding blood. So his initial thought is because his fall damaged his kidneys, that there was something seriously medically wrong with him. And we had to therefore confess what happened and what we did. And being the friends that we are, he goes, "That's really cool." So Bill was very impressed with what we accomplished, and he got us back various ways going through. But the thing about this particular drug, the rifampicin, is the kidney doesn't know what to do with it. It's like, I don't know what to do. Do you know what to do with that? They can't take that antibiotic. It doesn't recognize it, won't move it back into the blood. So as it moves through the kidney, it's just left in the nephron, moves to the collecting tubule, and ends up in the blood. And because of its color, it can be frightening to see. So, what is saved by the kidneys? There are transporters, there are active systems that will save a number of different things in the kidney. So, you can see here we've got bicarb, water, urea, glucose, amino acids, vitamins, proteins. All these things are going to be recognized by the nephron and moved back into the blood. Hey, this stuff is good. We need to save it. We need to put it back in the blood. Sodium, potassium, acids, magnesium, calcium, chloride, water. I think these are all repeats over here. Different sections of the nephron, proximal convoluted tube, loop of Henle, distal, collecting duct, collecting tubule, all move different things that need to go back into the blood. What doesn't get collected, so that antibiotic does not get collected. There are no transporters for that. There's nothing in the nephron that recognizes that as essential. And that antibiotic ends up moving through the collecting tube and down in the bladder. That tends to leave through the kidneys, stuff that's excreted in the kidneys. So you're going to have ammonia, urea, acids, and bases if we have too much of either. We feed our cows some acid to manage that milk fever. We're going to acidify the urine. The body says, well, it's in here. I don't need it. It's extra. It's going to acidify the urine so that extra antibiotic salts are going to go out that way. Acids, ammonium, urea is big. All those things are excreted along the way. And they're going to end up leaving the body, being excreted through the kidneys. Another diagram here shows sort of how the salts move, the electrolytes, and then the effect of two hormones. We have aldosterone here and ADH, antidiuretic hormone, which we'll see in the next slide. So aldosterone from the adrenal medulla, the adrenal gland, is going to help with our minerals. So we have sodium and chloride here. The hormone is going to affect this part, and it's going to add or promote the movement of our salts, sodium and chloride, across. So this is a way we can conserve those two electrolytes. They're still going to move over or be collected in a number of different areas, but aldosterone helps us conserve it, a little bit more active motion. You'll notice here we start out at 300. That should be recognized as an isotonic number. The cortex is more or less isotonic. The 300 exists in a number of different areas. Bowman's capsule, proximal convoluted tubule. As you go down into the medulla, the osmolality goes up, both in the tissues and inside the loop. Water is drawn out, especially going down. Electrolytes are pumped in. Water wants to move from a high to a low, so it's going to move down its concentration gradient, leave here, and then as it goes back up, you're going to have sodium chloride actively removed. We're going to reduce the solutes as we go up, and we're going to get back to that isotonic level here in the distal convoluted tubule. Now, depending what happens on the descending loop of Henle, or descending convoluted tubule or collection tubule, what happens to water here will determine whether or not the urine is dilute or concentrated. So we talked about water moving freely following the solute here. This more or less how things are set up. Over here, we have a section, this dotted section here is a space where water permeability is dependent on something else. And in this case, it's dependent on antidiuretic hormone. If the hormone is present, as we see 1, 2, 3, 4, the water moves out. If the antidiuretic hormone is not there, water does not move out. We get diluted urine. But in this case, it's present. Water is moving out following solute. We concentrate. We have an osmotic urine of 240 milliequivalents. Looking at the collecting tubule here, collecting duct is what it's called in this diagram. We have our hypotonic contents of the nephron coming over into the collecting tubule. We're moving from the cortex to the medulla. If antidiuretic hormone, no antidiuretic, ADH, it's also called vasopressin. If that is not there, this collecting duct, this collecting tubule is impervious to water. Water cannot move out. It can't flow down its concentration gradient. We got osmolality very high here. Water wants to move out, but the collecting duct, in the presence or the absence of ADH, will not open. When antidiuretic hormone is present, these walls will open up. Water will be absorbed back into the medulla. It'll move to the blood, and we will have more water being conserved in the blood. So this might be a situation where we're consuming large amounts of liquid. We need to get rid of them. The posterior pituitary in the brain does not secrete this. We've got lots of volume that needs to be excreted. So we've got lots of dilute or hypotonic urine being created, low in solute, high in water. If we're doing a lot of sweating, if you're like me growing up stacking hay in a haymow, very high temperatures moving through, you're going to want to conserve as much of that water as you can. You're running down every chance you get, filling up on a water hose, running back upstairs. that ADH, your brain tells you that the blood is too concentrated. You need to conserve water. So it tells the kidney to put the water back in the blood, opens up the collecting tube. So on a hot summer day, you're drinking a whole lot of water, but very little comes out through the bladder. So you have a hypertonic urine, very little water, high in solute. So we have a situation here without ADH, we have the opposite of our cheer. Water does not follow solute unless ADH is present. So no following solute here, following solute over here when ADH is present. So you should be able to tell me how the kidneys work, or what the kidneys do. We are trying to maintain the blood, and the nephron takes all the plasma out, pulls back everything it likes, and we reconstitute the blood. Everything that's waste, everything that's extra, everything that doesn't belong ends up in the bladder. That's the waste product of the process of what the kidneys do. The nephron moves solutes actively, water follows passively where it can. ADH needs to be present for that to happen. So this brings us to our discussion of hypertonic saline. We kind of alluded to it before, but let's make sure we understand it in the context of the kidneys. Water intake is determined by dry matter intake, milk production, and we're going to focus on the moment for sodium intake. There are sensors in the brain that measure the amount of sodium in the blood. As the sodium increases, we have a strong desire to drink. That's one of the reasons we have pretzels and salted peanuts and bar. We want you to buy more beverages to keep that thirst away. So sodium sensed in the brain says, hey, the blood's thickening up. The best thing I can do to dilute that concentration out is to consume more liquid, more water in the cow's case. So we take that hypertonic saline and we use it to treat environmental mastitis. Now, one of the problems with environmental mastitis is our white blood cells or leukocytes get in the mammary gland and they destroy the bacteria. So we talked about the white blood cells engulfing the the bacteria. They're going to secrete all kinds of enzymes. They're going to blow that bacteria up and then spit out the contents or the pieces when that bacteria is destroyed. Now, once that leukocyte spits out the contents or what's left over of the bacteria, we're creating a lot of things that make the animal feel very bad. So in this case, the white blood cells doing their job actually creates circumstances where the cow stops eating, stops drinking, feels real bad, runs a fever. All those bacteria parts are causing problems in the body. They're floating around there, they're sitting around there, and it's our job as managers to get rid of them. So those bacteria parts, we need to IV that hypertonic saline. We get that in the body. You're going to have, if you've ever done that, you have a 5-gallon pail right there. As soon as that blood or the hypertonic saline goes to the heart, to the lungs, and then out to the brain, it says drink immediately. So we're talking a minute's response. And they're going to consume that 5-gallon pail and maybe more, depending on how dehydrated they are. The idea behind this is now we have a big volume of water in the body. And the kidneys say, hey, that's more water than we need. And what happens is the kidneys open up, they take more of the blood flow. When when you're dehydrated, your kidneys say, well, maybe we don't want to work quite as hard. We're going to save the blood flow for the rest of the body. We'll not do our job. We'll get to it in a couple minutes when you drink. But now you got this big flood of water in. The kidneys say, hey, I don't need this. And they start to filter out large volumes of plasma. As they go through and they reconstitute that blood, the parts of the bacteria that are in that bloodstream are not recognized by the nephron. They are not pulled back into the blood from the nephron. So they're going to flow to the bladder and then leave the body. So, in effect, what we're doing with the hypertonic saline, the consumption of water, is flushing out all these bacteria parts that give that reaction that makes the cow feel bad, the fever, the anorexia, the just not feeling good, really off. We're flushing all those bacteria parts, those endotoxins out of the body because the nephron doesn't know what to do with them. I don't think these are good. We're not going to put them back in the blood. We're going to ship them off to the bladder. So that's how a hypertonic saline solution works. There's a article I put up on Blackboard that discusses that in more detail. You've got your hypertonic saline here. Again, we're putting a whole lot of salt, sodium and chloride into the blood to force that animal to drink, to flush her body, to get all those bacteria parts, they're called endotoxins, out of her body. And I believe that's it.